I cut off part of this slide at the end of the last lecture, so I thought we'd start up back here. Just kind of go over it quickly so we make sure that I didn't leave anything out. So as we were saying, these wrappers, these coverings, the epimysium, paramysium, and endomysium that are all kind of wrapped in different layers around, around and in the muscle, extend past the muscle on either end and form the tendon, which allows the muscle to be attached to the bone. So really, sometimes we hear about a pro athlete who their biceps brachii becomes disconnected. And in order for that to happen, it's a one heck of an injury because they're severing all of this massive connective tissue that's made up of strands. So it's really a catastrophic injury when it does happen. Now, an aponeurosis is a broad, flat tendon. We said that they would occur in to attach muscles to the skull where you don't necessarily, where a rounded cord-like tendon would get in the way, where you want something flattened, where that would work a little bit better, and that's the aponeurosis, excuse me. Each skeletal muscle is going to be supplied by an artery, one or two veins, and a nerve. And as we said, these skeletal muscles are highly vascularized. All of the muscles are highly vascularized. Excuse me. Coronavirus. Sorry, couldn't resist. Each skeletal muscle needs to have a really rich blood supply coming in to make sure that it's getting the oxygen and the nutrients that are necessary and a way to get rid of the garbage. So that's why we need the veins. We also need the nerves in order to send the stimulus that we talked about in those characteristics of the muscle. We said that it needs to be able to respond to a stimulus. It has to be excitable, right? So we want to make sure that we have a nerve supply coming in so that if these cells are excitable, we have something to excite them with. And I'm going to say this 100,000 times to you because it's a different way of looking at it. It's a different terminology that each muscle cell is referred to as a fiber. Not really sure why, but when you see the word muscle fiber, it's talking about a muscle cell, right? It's not any kind of a protein fiber or any of the other things that we've talked about that would be fibrous. It is, in fact, a cell itself. Now, this is a nifty diagram. This is going to show us the epimysium, the paramysium, what a fascicle looks like, and the endomysium. And again, that they're coming off of the end of a muscle and attaching the muscle to the bone in a very, very secure manner. When we look at a skeletal muscle cell, they've got some differences, okay? They're arranged differently. They have different components than the garden variety quote-unquote cell. So what a skeletal muscle cell is going to have in it is going to differ wildly from maybe a squamous cell or garden variety adipose cell. Again, to start with, and I've got it bold this time because I can't stress it enough because I know this is a tripping point for us. When we talk about a muscle cell, we're talking about a muscle fiber. Primarily because these skeletal muscle cells are larger than most cells. We said that they are multinucleated. There's an evolutionary theory that thinks that several more simple skeletal muscle cells kind of merged over time and formed this supercell. That's why there are many nuclei, and that's why it's so large, that it was in fact a merger of a whole bunch of skeletal muscle cells into one single cell. We need to talk about the sarcolemma. Now, this is important that we get. We are going to have a root, sarco, that we're going to see a lot when we talk about muscles. Skeletal, cardiac, smooth, doesn't matter. Sarco is going to become a recurring theme. Sarco refers to a muscular substance or flesh. So that's why we're going to see a sarcolemma, we're going to see sarcoplasm, we're going to see a sarcoplasmic reticulum, because it's referring to flesh, and most times when we're talking about the flesh of an organism, we're talking about the muscle. So the sarcolemma is a fancy name for the plasma membrane of a skeletal muscle cell. It's not any different. It's the phospholipid bilayer with our proteins jammed in there, just like we're familiar with. All right. Well, why has it got a different name? Because again, it's got a better PR firm. It's got a different name because it's specific to skeletal muscle cells or skeletal muscle fibers. Sarcoplasm is the cytoplasm contained within a muscle cell. It's a little bit different. All right, because the muscle cell is so specialized and it has such an important job, whether it's skeletal, cardiac, or smooth, the sarcoplasm is going to be a little bit different. It's going to contain glycogen. Now I'm going to, if we were together, I would say, okay, what's glycogen? So give it a think. Hopefully you're thinking, well, it's a carbohydrate. You're right. It is a carbohydrate. It is a polysaccharide. It's made of hundreds and hundreds of glucose molecules bonded together through dehydration synthesis. You'll remember when we talked about our carbohydrates, and again, your exam 1 packet is invaluable to you at this point. Always refer back and see what you wrote about it, see what you know about it to remind yourself. Well, when we did glycogen, when we were talking about our carbohydrates, we said it was how an animal cell stores excess carbohydrates, excess energy, right? Because remember, when we said we wanted to make ATP, we needed lots and lots of glucose. Glycogen is kind of a savings account of glucose for us. We said we were going to find glycogen in 2 specific places, in the liver of an organism and in a muscle cell. So here we are talking about the muscle cells, and here we are referencing our old friend glycogen. This is where it's going to be stored. It makes sense to have carbohydrates there. Why? Because muscles need lots of ATP. In order to make ATP, what do we need? That's right, we need glucose. So glycogen is going to give us a ready supply of glucose by breaking down glycogen through hydrolysis. See, you're never getting away from it. It's going to keep following you. It contains myoglobin. Myo, again, we know is muscle. Globin, hopefully, is going to make some of you think of the word hemoglobin. Hemoglobin is found where? In your red blood cells, that's right. So the job of hemoglobin in your red blood cells is to attach to oxygen and carry it, transport it through your body. The job of myoglobin is very similar. When oxygen enters a muscle fiber, it attaches to the myoglobin holds on to it because, again, muscle fibers need lots and lots and lots of ATP. What do we need to make ATP? We need glucose, we need oxygen. Myoglobin is going to store that oxygen. So the muscle cell has a bunch of oxygen right in-house, right in place, waiting for it to be needed, rather than waiting for a shipment, waiting for a delivery to come through the circulatory system. The other thing we're going to see lots of is many mitochondria. Again, refer back to exam 1. When we were talking about mitochondria, we said that we're going to have varying numbers of mitochondria depending on what the job of the cell was. And you'll remember, one of our examples was adipose, and they had very few mitochondria. Our other example were muscle fibers, saying that they had lots of them. Because again, I need to make ATP, because that's what my muscle fiber needs to do its job. If that's the case, what are the 3 things that I need? I need glucose, I need oxygen, and I need mitochondria to process those materials so I can get ATP. Well, glycogen is going to give us the glucose. Myoglobin is going to store the oxygen on site for us, and the mitochondria will take those raw materials, break them down, and supply the muscle fiber with lots and lots of ATP. We also have transverse tubules, they're referred to as T-tubules. Transverse tubules are going to be positioned transversely. Keep in mind, what does a skeletal muscle fiber look like? It's a long tube. It's that striped snake, It's the striped worm that we talked about. Transverse tubules are going to be positioned transversely or cross-section. They're going to go across the long axis, okay? Their job is to penetrate deep into the cells. They are an extension. They are continuous with the sarcolemma. So the sarcolemma is going to be jutting down deep into the muscle fiber by way of these transverse tubules, all right? It's named differently because it's in a different position, but it's continuous. It's the same material that we're going to see, but it's positioned deep in. I've got a diagram for you in a sec. The transverse tubules are critical because they're going to allow a nerve impulse to be conducted deep into the cell. Because remember, we said, what do we need? What were the characteristics of our muscles? That they needed to be excitable and they needed to be conductive. So we have to have a way to get that nerve impulse deep into the cell, and that's what the T-tubule is going to do for us. Now, looking at this diagram, the yellow tubes are the T-tubules, and you'll notice that they are positioned transversely going across the long axis. You will notice that that orange rind, if you will, that's been peeled away, that's your sarcolemma, and you'll notice that the yellow tube is continuous with that. Okay, so those yellow tubes that are going to be positioned along the length of our muscle fiber are the T-tubules. You'll notice in there it looks like a loaf of bread tucked underneath the bottom of that cell. That's one of our mitochondria. You'll notice there's another one kind of up towards the top on the far right side. To me, it looks like a loaf of bread. Maybe I just want to eat bread. I don't know. But those are our mitochondria, and you'll notice that there's lots of them tucked in. Once you see one, you're going to see several of them. The other thing I want to make note of is that purple kind of Swiss cheese looking stuff that's suspended between the two T-tubules. That purple material is the sarcoplasmic reticulum, which we're going to talk about next. Again, sarcoplasmic reticulum hopefully is going to make you think of the endoplasmic reticulum. This is a special kind because it's found in a muscle fiber. That's why we have sarco in there. It refers to a specialized version of the smooth endoplasmic reticulum that is there to store and release at the appropriate time calcium ions. As you're going to see in subsequent lectures, calcium ions are crucially important to how a muscle fiber is going to contract. So we have to have a spot where we can store them so they're readily available to be released when necessary. As we saw, they're going to be positioned perpendicular to the T-tubules. They're going to be suspended between the T-tubules. We also have myofibrils. These are the contractile proteins within the cell. These are protein molecules. This is why, this is how a skeletal fiber, muscle fiber, can do its job. These myofibrils are going to be what causes a contraction to happen at the molecular level. They're arranged in something called sarcomeres, which we'll talk about. There are two types, thin myofilament, or the actin molecule, and thick myofilament, or the myosin molecule. You may want to take a moment before we go too much further and maybe read a little bit in your